So we're on to Maths Skills Homework 8, log graphs. Lots of fun with these. And why do we have log graphs? We have log graphs because our data has got a very large range, and it enables us to plot the data accurately and draw lines of best fit on much better. So plot this data on the graph. Well, we've got, let's put our axes labels on first. Age of culture along the bottom, so that's our, you could do this in pencil, so you can rub it out, and you can always use pencil, nice sharp HB pencil. I'm going to do it in red pen just to make it stand out, so I hope I don't make any mistakes. Age of culture hours. And I'm just going to copy this title. I can't get it wrong if I copy that title. And on the side here of the X, our dependent variable, which is number of cells, mean of 5 replicates. I can't get that wrong either. The scale on the bottom here is pretty straightforward. I'm just going to 1, 2, 3, 4, 5, and I know later on it's going to ask us to do some more, so I'm going to stick those on. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Up the side now, this is a tricky one. If I put my little lines on, you can see this graph paper is log paper, which means the divisions change, gradually get smaller, then another cycle begins. So if I identify these for you, they should help you with your plotting. So that's one sort of cycle, and then it does the same thing. And it starts off at 10, so the next one is going to be 20, 30, 40 and so on. So 20, 30, 40, 50, 60, 70, 80, 90, 100. So you can see they get squeezed up. And then where's the next one? It's there. And then the same thing. So we're looking for a slightly smaller one, slightly smaller again, and so on. If I've done that right, yes. Apart from this time, it's not going to be 110. It goes to, the scale changes. It's 200, 300, 400, 500, and that's why it's a log graph. It's a logarithmic scale. 600, 700, 800, 900, and we end up with 1000 at the top. So at the bottom of the graph we can go between 10 and 20, but at the top of the graph that's almost disappeared, and it's a whole 100 in a small space. And then we just need to plot our points. So at 0 hours we've got 50. So I might do this in another colour for the points. There we are, there's my blue pen handy. So at 0 it's 50, there we are. I'm just going to put a nice clear cross on there. You could do this in pencil, of course. 1 is 60. So 1 is 60. You could plot these as a dot with a circle around, or a cross. I'm using crosses. 2 is 72. So there's 70, and there's 5 little squares, so I'm going to put it on the first little square. Pretty tricky to plot. 386.4. Yikes. So that's 80, and each one, so 82 for those there somewhere. Pretty difficult to plot that. 4 is 103.6, so 100. Well, this next one is 110. So 103 is going to be sort of there. And 5, 124.4. 120, so that's 100, 110, 120. Next one is 130, so you want about a quarter of the way there. And there's our point there. And in biology, we ask you, if we just got a single set of points, just to join them up dot to dot with a ruler. Or these are actually suspiciously all in a straight line. And that's why we've used log graph for this, because with it being an exponential rise, so an exponential rate, if we had a normal graph, it would sort of start off slow and then up like that. Then it turns that sort of a line into a straight line. So we can, on this occasion, draw a straight line through these and extrapolate. So there's our line, dot to dot through the points. And then it says extrapolate, carry on the graph using a dotted line. So now I'm going to use this dotted line to carry on. So I'm going to use this dotted line to carry on, just like it says in the question. Okay. So that's our graph. That doesn't look too bad. And we've got a number of cells at 7 hours. So for this, I think I will use a different colour again. So that one I've got. Someone's hiked off with my my green pen. I'm going to use my. I'll just use the blue pen again. And 7 hours. So I'm just going to draw a line up from 7 hours. And if you can get this bang on 7, then that's brilliant. Okay, up to the line, and then I'm going to then draw a line going across from there. Okay, and then you can read off on that scale whatever you've got. So on my line, that's going to be about 110, 20, 30, 40, 50, 60, 170, about halfway between 170 and 180. That's going to be 175. So mine will be 175 there. So 7 hours equals 175 cells. And probably there'll be a little bit of leeway here, between 170 and 180, maybe a little bit more for slight variations in that extrapolated line. And the last one, it says 14 hours. Well, it's easy to draw the line because it's just bang on the end of the graph. So it would be that one. And it's always good to put these lines on, then who's ever marking it can follow where you are and what you're doing. And again, I'm just going to draw that line going across. So on mine, it's probably the first little division. So on mine it'd be 620. So 14 hours equals 620 cells. There's a mean number of 5. I could write all that out there. But again, you might have done the line slightly differently. So anything sort of 600 to 640, or even a little bit beyond. If they can see what you've done, that would be acceptable there. And then there's a last line to this. It says, do you think the extrapolation is justified? Give a reason for your answer. Well, first of all, we could answer yes. And if you've answered yes, you can go, yes, it is justified because look at my points, they're in a wonderful straight line. It's a reasonable thing to say that they would carry on going in a straight line. And it's reasonable to suggest, certainly for 7 hours, that 175 would be the number there. That's a reasonable suggestion. So yes, it's a straight line. So good for making a guess there. How about no? Could we answer no? Well, at 14 hours, this is a culture and where we're growing something. Well, at some point, these cells are going to stop being made because we might run out of food in the culture, might run out of oxygen in the culture, and there might be just not enough room in the culture for all of these cells. So there might be some other problems. They might produce toxins, or the culture might get contaminated. All sorts of reasons why this culture might not carry on growing forever. So no. For example, the culture may run out of food, or next year when you do that, you might say substrate or suggest whatever food it is. So the answer there, no marks for yes or no, but the mark for the actual justification. Give a reason for your answer, which some people missed out. And so hopefully you can now draw and use log graphs.